start with a very important topic, okay, which is photosynthesis in higher plants, right? So we'll start the chapter. Now we have already, what is a photosynthesis, right? It is nothing but what the green plants is going to make the food by themselves using some of the raw materials, which includes carbon dioxide, water, then what else? Chlorophyll in presence of sunlight, and they provide what? They provide the glucose, which is required for the growth of the plant. And animals, we are including human beings and all, they are actually depending upon whom? The plant for food materials, right? So photosynthesis, the chapter begins with experiments. So we will take up those experiments. So the first experiment, it is about experiment to prove the essentiality of raw materials. The essentiality of raw materials. So we already know what all are the raw materials required for the process of photosynthesis. So here we will take up the essentiality of, carbon dioxide. We will take up the essentiality of chlorophyll. Also we will take up the essentiality of sunlight. Right. So first we will take up the essentiality of sunlight and chlorophyll. Essentiality of sunlight and chlorophyll. We are taking a cotton plant and the peculiarity of the plant which we are taking is it is a plant with variegated leaf. Let's say it's a plant and this leaf we are saying it is a variegated leaf. What understand by variegated leaf? This leaf, it is having some yellow patches. It is green in color, along with some yellow patches. So what we are doing is, one of the leaf we are covering with what? A black paper. We are covering with a black paper, and we are exposing this entire experimental setup is illuminated under sunlight. Later on, the leaf which is covered is tested for starch. We know the iodine test is tested for starch, and in that test, it has shown that the part which is covered, it gave what? Negative test. Or it gave the negative result, indicating starch was not formed in the covered region. We know that by the process of photosynthesis, what we form carbohydrate, and the carbohydrate is accumulated in the form of starch. So in the covered region of this particular plant, there was no formation of starch, which indicates photosynthesis did not happen in the covered region. Along with that, when the another leaf, which was non-covered, when it was taken for the again, we can say negative result it gave some negative result in the yellow patches. Because we have taken a variegated leaf, so we will know variegated leaf is having yellow patches. So in the yellow patches, there was no formation. It proves what? Chlorophyll in that region, and hence photosynthesis did not happen. So we can conclude one thing. For process of photosynthesis, sunlight is also required and chlorophyll is also required. Now we will move on to the second experiment to prove the essentiality of carbon dioxide. Essentiality of carbon dioxide. So this experiment we call as Moll's half-leaf experiment. Moll's half-leaf experiment. So again we are taking a potted plant. Now, to this potted plant, the leaf of this potted plant, it is actually inserted into a jar. Okay. Inserted to a jar, and it is completely insulated. Half of it. And this jar is actually containing a cotton that is soaked in KOH solution. KOH solution. So KOH is having one peculiar feature. What is that? It will absorb carbon dioxide. So what will happen? The region inside the jar is now completely away from carbon dioxide. Carbon dioxide is completely absorbed by this KOH. So we can say half of this particular leaf is now exposed region where there is no carbon dioxide at all. After some time, we are exposing the experiment step under sunlight, illuminating it under sunlight, and again we are going to the start test. So after the start test, it was clear what happened when they took the leaf. Half of the leaf which was not inside jar gave positive result for start test, and the region which was inside the jar gave negative result. So what does that mean? The region which was inside this particular jar did not do photosynthesis due to the absence of carbon dioxide. And why there is carbon dioxide? The available carbon dioxide was absorbed by KOH. This proves the essentiality of carbon dioxide. Now we'll move on to the historical background in photosynthesis. So the first one, it is the experiment of Joseph Priestley. Experiment of Joseph Priestley. So Joseph Priestley in the year 1770 conducted a very famous experiment known as the bell jar experiment. The bell jar experiment. So what is that experiment? It is nothing but including a jar which is of bell. So he took a jar in the shape of a bell. Okay, and into this jar, first he introduced a burning candle. A burning candle was there. So number one, bell jar plus burning candle. A bell jar and a burning candle was taken. After some time, what happened? They found out the candle was extinguished. So what they found the result? candle was extinguished. Now, later, he took another bell jar, and into that bell jar he introduced a mouse. And what he observed? Mouse was dead due to suffocation. Mouse was dead. Okay, so he took a mouse also. Now, later, what he did is he took a bell jar, and into that he kept a burning candle along the mouse, and he also introduced a plant. So what did he observe? He observed that the candle did not extinguish. Mouse was alive. Okay, so introduce into this. So what he concluded from this experiment? So Joseph Priestley, whenever he took a candle along, result it was extinguished or dead. But when he introduced the plant, the candle was burning as well as the mouse was also alive. So he hypothesized. He hypothesized one thing. He said that the burning of candle and respiration of fat, it produces a dirty or foul gas, and he called this dirty or foul gas phlogiston. Phlogiston. Okay, dirty or the foul gas as what? Phlogiston. And this gas was purified by the gas which was produced by the green plant known as D-phlogiston. D-phlogiston. Okay. Burning the candle and the mouse, respiration of the mouse, it has produced a dirty which was purified by the gas. The dirty gas he called as phlogiston, and the good gas purified because he called it as D-phlogiston. So now that what is phlogiston and phlogiston? Phlogiston is nothing but the and D-phlogiston is oxygen, right? And in the on to the next experiment. Experiment of Jan House. So Jan Engine House also have a similar kind of experiment like Joseph Priestley. have an agreement to Joseph Priestley. What was that? But the Belger experiment actually hid into conditions. Under two conditions. Yes, sorry for the interruption. There was a network issue. So we were discussing about the experiments, the historical background. So we were discussing about the experiment of Joseph Priestley. So Joseph Priestley's experiment, what he did is he took a bell jar. In the bell jar, he took a burning candle, and the burning candle, after some time, it was extinguished. Later on, he took a rat or a mouse, and after some time, he observed that the mouse was dead. Later, he have taken a mouse and introduced a plant into it, and he found out that the candle was burning, not at all extinguished, and he saw the mouse was dead, sorry, was alive, and the plant was also there. So this proved that the burning of candle, he hypothesized the burning of candle and the respiration of the rat is producing a foul gas which he calls phlogiston, and this phlogiston was purified by the gas produced by the plant, phlogiston. Right now, we were moving on to the experiment of Jan Ingenhouse. So he did a similar experiment which was done by Joseph Priestley. What was that experiment? He took a bell jar, the same bell jar with the same experimental setup. What was that? A bell jar having a candle, a rat, and a plant. Now he kept this bell jar under two conditions. One, he kept the bell jar with organisms inside in presence of light, and another one in absence of light. So we very well know that the plants will do the process of photosynthesis. So in absence of light, this experimental, in absence of light, this experiment showed that candle was extinguished. The candle was extinguished and the mouse was dead also. Later, in presence of light, the same old result. The same result has come. What's that? The candle burned, and mouse was alive. So he also said that the purification of the gas by the plant, that is the conversion of the phlogiston into phlogiston, it will happen in presence of light. Okay. Another very famous experiment which was done by Jan Ingenhouse was on a water plant. So there what he have taken, he took a beaker. It was filled with water. To this beaker, a water plant was introduced. Water plant, the plants which live in water, water plant. Okay, and a funnel, an inverted funnel was also introduced. One plant like Vallisneria and can be taken, and this is the beaker containing water. Okay, now after some time, he was able to see there were some bubbles moving out of the funnel when it was illuminated under sunlight or under normal light. Okay. When it was illuminated under sunlight, he was able to see what the bubbles moving out of the funnel, and this later the bubbles of oxygen found out. So he concluded that this Process of photosynthesis produces oxygen as the byproduct. Oxygen as the byproduct. Okay. So we will move on to the next experiment. Experiment of Julius von Sack. Of Julius von Sack. So Julius von Sack, that experiment detail part we need to see, pointed out the green part of the plants. Produces glucose. The green part of the plant, it produces the glucose. Now, by the 19th century, that's all about the experiment. By the mid-19th century, the raw materials which was required for the process of photosynthesis was what was known. So they included water, carbon dioxide, and the empirical formula of the photosynthetic process was also made by the mid-19th century. So what they said, the carbon dioxide combines with water, gives you carbohydrate along with oxygen as the byproduct, right? In presence of light. So at that time, it was formed as the byproduct. So people or the scientists at that time assumed that the oxygen is liberated from the carbon dioxide. Okay, it was believed that it was liberated from where? It was liberated from the carbon dioxide. But now we know that it is from the water, but at that time it was not known. So we will see what exactly or what are the experiments to prove that water is the electron donor or photo is the water produces oxygen. Experiments to prove evolution of oxygen by photolysis of water. By the photolysis of water. 3 experiments are there. Experiment of Van Niel, experiment of Hill, and experiment of Rubin. 3 experiments. Okay, experiment of Van Niel, and experiment of Rubin. So we will take up the experiment of Van Niel. Van Niel actually have taken sulfur bacteria as an experiment organism. Sulfur bacteria, you have already studied the classification. The photosynthesis of sulfur bacteria, they also utilizes carbon dioxide. Along with carbon dioxide, it also takes the what? The sulfide containing H2S. So here H2S is the proton electron donor, because we know that in the process of photosynthesis, it is water which is going to be the electron donor in the ETS. So here the electron donor is going to be H2S, not H2O. Water is not, no water at all, not that point. And as per scientists, if carbon dioxide is the one which is responsible for oxygen evolution, here carbon dioxide is there. So definitely here, along with glucose, what should be there? Oxygen should have been there, but here oxygen was not there. Here, instead of oxygen, sulfur was there. Okay, S was formed there. So what we can assume from this, we can say that S2, sorry, S was formed here, right? So we can say the compound was the electron donor here, and as a result we got the sulfur compound as a byproduct here. If carbon dioxide was the oxygen reducing factor, here even though carbon dioxide is there, we did not get what? Oxygen. So carbon dioxide is not the responsible factor according to this experiment. So this is the experiment which is given in your NCERT in little bit of detail, and another two experiment we'll just go through the what organisms used. Second one is experiment of Robert. In the experiment of Robert, he have taken still area, chloroplast of still area. And next one is experiment of Reuben. So Reuben have taken a unicellular algae. I will say this experiment, Reuben's experiment is very simple. What he did is, he have taken what? A chlorella and given water containing radioactive oxygen and carbon dioxide. So when he observed, there was radioactivity in the oxygen which was released as a byproduct. So it has proved that water is the one which is involved in the evolution of oxygen. Clear? The water which was provided was radioactively labeled oxygen, and the carbon dioxide was having oxygen. So at last, oxygen will be produced as a byproduct, and the oxygen was tested for radioactivity. So we provided radioactivity in water. So ultimately, at last, it is the oxygen which came out with radioactivity. So clear? So this was the experiment of evolution of water by the photolysis of evolution of oxygen by the photolysis of water. Now we will move on to the site of photosynthesis. Where does this exact photosynthesis takes place? Site of photosynthesis. So where exactly does the photosynthesis takes place? Since lower classes we know that it takes place in green leaves. We have taken the experiment in the beginning of this class. And in the green leaves it takes place where? In specialized cells. The name of the cell is mesophyll. And in mesophyll cell, it takes place where? In specialized organ known as chloroplast. So that is why chloroplast, they are considered as the kitchen house of the cell. It is known as the kitchen house of the cell. Now we will take up the structure of chloroplast. Already the structure of chloroplast is on to from the cell chapter. Still we will see the structure. Chloroplast is usually discoid or oval-shaped cell organelle, and it is having a double membranous structure. A double membranous structure. So one more membrane. Double membranous structure. Now, so this is going to be the outer membrane. And this will be the outer membrane, inner membrane. And inside of the membrane is filled with a jelly substance which we call as the stroma. Stroma, right? Stroma is present. Stroma is having certain structures present inside them, which is formed from the membrane, which we call as the thylakoids. Thylakoids. Now, these thylakoids, they are not seen as what? Single structures. They're actually found one above the other, just like stacked of coins. Right, stacked one above the another like this. These stacks of thylakoids, we call it as the grana. The grana that are interconnected to each other. By means of a structure known as the stroma lamellae or fret lamellae. Stroma lamellae or lamellae. Now, within the stroma lamellae or within the stroma is also having a circular DNA, which we call plasmid. So, due to the presence of DNA in the chloroplast, we say that this chloroplast it is a autonomous organelle. Semi-autonomous cell organelle means what? It need not to depend upon the nucleus for all the functions. Some of the functions they can do by themselves. So the structure of the chloroplast, where the process of photosynthesis will take place. Now, thylakoid is actually having various photosynthetic pigments, and this photosynthetic pigments involved in what? Will capture the light energy, and as a result, it will produce some of the assimilatory powers like ATP and NADP. So we will come to that. So that process of ATP production and NADP production using light energy, we call it as light reaction. So thylakoid is a place where the light reaction of photosynthesis takes place. Along with that, the stroma has various enzymes which is which is helping in the process of dark reaction. So that dark reaction is also known as stroma reactions. So stroma is a place where dark reaction will take place. So two things. Thylakoid is a place where a light reaction is happening, and stroma is a region where dark reaction is happening. Okay. Now we'll move on to the photosynthetic pigments. photosynthetic pigments. So the photosynthetic So coming to the photosynthetic pigments. So there are three major. We are studying about photosynthesis higher plants. So in that particular higher plants, if you are taking, there are three photosynthetic pigments, which includes chlorophyll, carotenoids, carotenoids, and phycobilins. So chlorophyll, you know that is green colored, and we are having different types of chlorophyll, like chlorophyll A, B, C, D, E, etc. Carotenoid, it is orange yellow colored. Okay, and we are having two types, carotene and xanthophyll. Carotene and xanthophyll. And phycobilins, it can be blue or red color. And again, 3 types of phycobilins are there. Phycocyanin, allophycocyanin, and phycoerythrin. And phycoerythrin. So, 3 types there. Okay. Now, detailed part of this one is not required now. We will move on to another topic, that is absorption spectrum. Absorption spectrum. Absorption spectrum. So absorption spectrum, we can say, is nothing what it is a graphical representation of degree of or amount of wavelengths. Okay, it's a amount of wavelength which is absorbed by a compound. Here we call that compound what? That's what our pigments. So the amount of wavelength, the graph which is going to represent the amount of wavelength which is absorbed by the pigment molecules. Okay, so that it is graphical representation it is a graphical representation of degree or amount degree or amount of wavelength of light wavelength of light absorbed by a and we know the resistance is the pigment. The pigment going to the substance. Now we take in this chapter, as per the NCERT, we have to take up the absorption spectrum of 3 pigments: chlorophyll A, B, and carotenoids. Chlorophyll A, B, and carotenoids. Okay. So we will take up the graph. Okay, this is the graph. The wavelength of the light, and this is the rate of absorption. Now, (400) in nanometer. Okay. Now, chlorophyll A, it shows a maximum absorption. It has maximum absorption 2 regions. One in the blue region with the 435 nanometer, and as well as in the red region 673. 435 nanometer and 673. So 435 is to be blue region and it is going to be in the red region. So maximum absorption is shown by them. So we will draw that 430, 673. And we will mark it as plus A. Now for chlorophyll B, it shows again at two regions. Chlorophyll B will show a maximum of 453. That is in the blue region, and in the red region was 642. 642. Four and 53, 642. This is and next one is about carotenoids. Carotenoids option is very close in places at 498 and 499. $498 and $499. So the graph is very important. So that they can give the graph as such and ask you to label which one is more or which one corresponds to the pigments. So if you absorption maximum at 435 and 673, it will be chlorophyll A, and another 450 and 642, it will be for chlorophyll, and 78 499 is for carotenoids. So this is the graph which is going to represent the amount of wavelength of light, the pigment molecule A, B, and carotenoids. Okay. Now after this we will point if you are comparing both chlorophyll A and chlorophyll B. It was found that chlorophyll A is having more absorption in the red region, and chlorophyll B is having more absorption in the blue region. So also switch. Now next one we move to the topic is action spectrum. Action spectrum. So previous was absorption spectrum, action spectrum. It graph representing the effectiveness of the wavelength, which means by this wavelength or which wavelength is for process photosis. So it is a graph. It is the graph representing the effectiveness of different wavelength. Performing a function. Performing a function. And here we know that what is that function? The function is photosynthesis. Okay. So we are going to show it maximum rate of photosynthesis is done. So the rate of photosynthesis can be analyzed by one factor. The rate of the photosynthesis is actually analyzed by the amount of oxygen liberated. So more photosynthesis means will be amount of oxygen released. So we will take up an experiment from. So it was studied by the scientist England. Studied it together. So he took organisms like Spirogyra and Chloroplast. Along with Spirogyra, also take oxygen-loving bacteria, which we call as the bacteria. So oxygen-loving bacteria means these are the bacteria going in the maximum oxygen evolution is there. So wherever maximum oxygen evolution is there, their growth will be to the air. Okay, oxygen-loving bacteria or aerobacteria. So we have this experiment done by Engelmann. Engelmann took a prism and reflected the light wave into what? Into a into color. G, or we know that. And prism, he actually scattered the lights, and in where light he have grown the spiral. Spirogyra algae, unicellular algae. The algae. Now, what will happen? Using this light, the algae will form photosynthesis. And also, he made a colony of aerotic bacteria, our loving bacteria to grow here. Okay, light was given. So what will happen? After some time, we said that using this light, they form the photosynthesis, and as a result, is a byproduct. So here, what will happen from here? From this, photosynthesis, oxygen, and that oxygen will be more in the blue and red region. It was found that it was more in the blue region. In those regions, the of oxygen loving bacteria was more as compared to other regions. Okay. Yes, so it was there, but more amount of oxygen loving bacteria was the blue and the red region. So this proved that it proved clearly that the more rate of photosynthesis or photosynthesis mostly happen in the blue and the red region. All of the bacteria was found where? Found in the blue as well as in the red region. Now we will move on to the next absorption period. So next one we have taken the absorption spectrum. We have seen the pigments showing various level of absorption. Then we have taken the action spectrum by the blue and red most useful for the process of photosynthesis. Now to the next is the immersion experiment. Immersion experiment. So here the effect of monochromatic light on different wavelengths. It's the effect of monochromatic light of different length on the lamp. On chlorella. So what was that? First one, what observed that the after 180 nanometer, there is a sharp decrease in the rate of photosynthesis. There is a sharp decrease in the rate of photosynthesis. He called this as what? He called this sharp decrease as red drop. Why? Because the 680 nanometer is almost in the far region. So there was a decrease in the rate of photosynthesis. This was that what the red drop, and also he studied the rate of observation. He made few studies. Okay, so he studied the rate of photosynthesis under some of conditions. Under the following condition. So first one, a shorter wave was made. A shorter wavelength, okay, 650 to 680 nanometer. 650 to 680 nanometer was provided. And what was observation? The observation was the photosynthesis was over. The rate of synthesis was poor when this particular wavelength was given, and a long wavelength was given next. A longer wavelength was given, and in the longer wavelength, say between 700 to 720 nanometer was provided. There also the photosynthesis was poor. Rate of photosynthesis was poor. Then he applied both wavelengths together to the chlorella. Supplied both, and observe there was 25% increase. 25% increase in the rate of photosynthesis. So we said that in the red region, when the wavelength or longer wavelength of light was given, the rate of photosynthesis, the increased photosynthesis we are getting back, or it is enhanced. That's why this is a conversion effect or enhancement effect. Emission effect or enhancement effect. Just graphically for our easy understanding. Rate of photosynthesis. So first case we have given 50 to 680 nanometer. We gave 700 to 720 nanometer, and both were given there is of increase. Okay, which we have of hand effect. So what he concluded? He concluded that the rate of photosynthesis is smaller, less small, larger, which is increased when given simultaneously. The rate of photosynthesis is less in small and larger wavelength of light, which is increased given simultaneously. Which is increased when given simultaneously. Simultaneously. Okay. So he also in this experiment he concluded that the plants are having two photosystems. Photosystem means it is nothing but the association of pigments. Come to the next topic. So plants have two photosystems. It is two systems. One will be activated at what we can say low intensity of light, and another will be activated at a high intensity of light. High intensity of light. High intensity of light. At one we call it as the photosystem 1. Okay, we call it as the photosystem 1. So one will be activated at a high intensity and another one at the low intensity. Okay, photosystem 1 and photosystem 2. Now we will take up the structure of photosystem. What exactly is this photosystem? Structure of photosystem. So the photosystem basically has two main parts. It has a reaction center light harvesting complex. And light harvesting complex. reaction center and light harvesting. Now, what exactly is a photosystem? We know that in plants there are different kinds of pigments. We are having different types of chlorophylls. Then we are having phycobilins. That also all the pigment molecules. What will happen? They all will join together and form a system, which we call a photosystem. So photosystem is an assembly or the association of all the pigment molecules. All the pigment molecules will join, and for every pigment molecule there is something which is very important, who is having most of the concepts. Just like a association is a president. So here the president name is reaction center. So the reaction center is nothing consisting of a single molecule of chlorophyll. Single molecule of chlorophyll A, and the light harvesting complex, it is what can consist of other pigments and proteins. And proteins. Okay, so we will just draw a diagram of a photosystem. So as said, it is having a reaction center, which is a single molecule of chlorophyll. It is all surrounded by various pigments. These are the accessory pigments. Okay. Now, so this is the reaction center. And this is the pigment and protein harvesting complex. So what will happen? Wavelength of light or the photons, it will actually trap the light harvesting complex or reaction centers, and what to do? This is passed, this light energy which has been captured by them, it has been passed to the reaction center. It's passed to the reaction center. So now the reaction center is getting very good energy. As a result, it will enter into a localization, during which what will happen? There will be a liberation of an electron. Electrons are released. The electron is released. This is complete structure of photosystem. So clear? So if the light harvesting complex will actually, it will get energy, and that energy which is light energy, which they are getting, it transfer to the reaction center, and from due to that now the reaction state due to an electron will be released out of them. Okay. Now, as I said, obviously that plants are having photosystem 1 and photosystem 2. Right. So what exactly is the difference in photosystem 2? Which is that one will be activated at a higher wavelength of light and another will be at a low wavelength of light. Okay, one will be activated at a high energy and another at a low energy or a low wavelength of light. So photosystem Okay. So here what will happen? It is actually getting activated at 700 nanometer. So that's why say 41 can be written as P700. Photosystem 2, on the other hand, is getting activated at comparatively lower wavelength, right? Around 180 nanometer. So that is why we are writing as P680. Now, let's say I draw a structure of a thylakoid here. Better to say grana. So let's say it's a grana. Now, this is a grana. This is thylakoid which is towards outer side, right? And also outer. The thylakoid is towards the outer side. We call it as the oppressed. So non-oppressed. Non-oppressed thylakoid, which were outside. And the thylakoid that is towards the region, we call it as what? So PS1, they are actually found where? It is found in the non-oppressed. Found in the non-oppressed region of the thylakoid. Along with that, it is also found in stroma lamellae. What was stroma lamellae? Stroma lamellae one connect granule. Whereas PS2, they are found where it is found only in the upper region. Oppressed. And the non-region. So non-oppressed means that is found towards outside, and oppressed means towards. So we want to process of photosynthesis. Okay, process of photosynthesis. So process of photosynthesis is basically takes place in, as we already said. Number one is light action. Number two is dark reaction. So one will be light dependent reaction, another light dependent reaction. Dark reaction does not mean that it is taking place during night. Reaction means it is independent of the light. It can even take place during daytime. Okay. Now we start with the light reaction. So light reaction again can be of two types. One will be the non-electron system, and we are having the cyclic ETS. Non-cyclic ETS and ETS. So what is the difference between one will be a